Hey guys, welcome to the Chapter 12 lecture. This will probably be done in parts. I don't know if I'll get through the whole thing before Lena disrupts us, but if she does, I promise I'll let you meet her. And Chapter 12, so your next test is going to be on Chapters 9, 10, and 12. Your last test in the class before the final will be 9, 10, and 12. 9 and 10 are relatively short. I combined them into one lecture, and it's a lot of conceptual sort of stuff. Same with Chapter 12. We've sort of moved away from all of the math in the course. We're really down to the biochemistry, really getting into the real meat of the biochemistry. So Chapter 12 is all about metabolism, namely the reactions that we use to break down our food particles and convert them into energy, ATP. It's there's sort of a lot of parts, there's a lot of steps in these reactions, and there's a lot of different reactions to keep track of, so it can be a little bit confusing or a little bit overwhelming, so I'll probably take my time on it. The lecture's fairly long, it's 58 slides, so it'll probably easily be a 2-hour lecture. And just review it if you need to, review the chapter, and definitely practice the problems in the chapter, because it's not, I don't personally think it's difficult material to conceptualize. It's just kind of a lot to hold on to. So I'll try to repeat myself a lot and, kind of get the repetition in there so you get it down. Anyway, this chapter is, so these are the sort of bullet points, the overview of the chapter. The first part, we're going to do an overview of metabolism. What is metabolism? What does that word mean? Then we're going to talk about metabolically relevant nucleotides. These are the molecules that capture energy from food and then allow us to convert it into cellular energy called ATP. And 12.3 will go over digestion, how sort of the overview of how all of our food molecules get broken down. Carbohydrates, fats, and proteins is what I should say, are macronutrients. Carbohydrates, fats, and proteins all get digested into their building block components, and then those building block components get broken down by different chemical reactions to form ATP. So glycolysis is the breakdown of glucose or carbohydrate building blocks. And the citric acid cycle is, so then there's several sort of pathways, metabolic pathways that occur in order to turn that macronutrient into ATP. It's not just like snap of your fingers and it's done. There's multiple steps and multiple pathways. And finally, we get ATP production here after going through glycolysis, the citric acid cycle, and the electron transport chain. Okay, 3 different metabolic pathways. And then the last part of the chapter talks about other fuel sources, energy sources. Okay, so metabolism, the word metabolism, is talking about all of the chemical reactions that occur in your body. It's the sum of all the chemical reactions that occur. And chemical reactions in your body occur for one of 2 reasons. Either it's to break down molecules, or it's to build up molecules, so to build proteins, to build cellular, like cell walls, okay, cell membranes. Or it could be to break down things, like to break down your food molecules, carbs, fats, proteins, or it could be to break down proteins in the cell that are, not working properly, or if a cell gets old and it dies and you want to break it down and sort of recycle the parts, okay? So all of these reactions occur. It's not just like one reaction. Like when we talk about chemical reactions, we have our reactants and our reaction arrow, and we have products. Okay, it's not, it looks like a one-step process, but with metabolic pathways, it's really, like a 5, 10, 20-step process to get a cell broken down into different components. It's not just one simple step. So these different types of reactions, if you're doing a chemical reactions to build up things, to synthesize molecules, we call that anabolism. Anabolism is the synthesis of like proteins in your cells, or DNA if you're replicating the DNA in your cells. Catabolism is breakdown, and that's really what we're going to focus on in this chapter. We're going to talk about the breakdown of biomolecules, specifically carbohydrates, fats, and proteins, those macronutrients from your diet. So anabolism, there goes Lena in the background, is a series of reduction reactions. So actually I have a nice little, from your textbook, the sort of slide here, okay? So catabolism is taking large biological molecules and breaking them down into their building blocks. That's what we do with digestion of our food. And it typically generates ATP. So when you think of bonds, okay, think of a bond like a stick, okay, like a pen. And if you break it, in order to break it, it takes energy, right? You have to input energy to break this stick or break this bond, okay? So it releases, oh, no, that's not what I meant. Never mind, I just got confused backwards. Well, anyway, when you break down your, I'm thinking about chemical reactions breaking bonds, but okay, so when you break a bond, you have to input energy, and then when you make a bond, you kind of store energy in it, and it depends on the bond that you make. So like if I broke this pen in half and then I tried to like tape it back together, that would not be storing very much energy in that new bond, because it'd be very easy to break if it was taped together. But if I like super glued it together or like welded it together, it might even be stronger than it was before I broke it, and therefore the new bond contains higher energy and is stronger, and so in forming those new bonds you actually are creating energy. But that's a little like more in depth. So I guess what I really want to simplify it to is when you break down large molecules, you break down, think of breaking down your food molecules, okay, you create ATP, you create energy. When you want to build things, okay, so think about building a house, okay, it takes a lot of energy to build things. In order to make larger molecules, you then have to use that ATP, that energy that you created from breaking down molecules. So these catabolism reactions are all oxidation reactions. Remember, I don't remember what chapter it was when we talked about redox reactions. It was the chapter on carbohydrates. I think it was chapter 6. So you might want to go back and review that real quick and remind yourself that hopefully it's still fresh in your head from doing the take-home exam. Oil rig was the mnemonic. So oxidation is loss of electrons or gain of oxygen. And reduction is rig. Reduction is gain of electrons or hydrogens and or loss of oxygen. Okay, so we'll see this when we look at some of these reactions, especially these catabolic reactions that we're going to look at. We've already looked at anabolic reactions, believe it or not. If you think back, we learned about how glucose monosaccharides combine to form disaccharides and polysaccharides. We're building larger molecules from a building block. That would be anabolism, the formation of glycosidic bonds, and that's a reduction reaction. Also, when we're forming peptide bonds between amino acids, again, another form of reduction, okay, an anabolic pathway. So these are our two forms of metabolism. So like I said, we're going to focus mainly on catabolism in this chapter because we've already sort of discretely done a bunch of anabolism. So catabolism of the biomolecules in our food looks something like this. This is the overview, okay? So this first level here, the first thing is that biomolecules in our food, proteins, polysaccharides or carbohydrates, and lipids all get broken down through hydrolysis reactions, okay? Water, they combine with water and get broken down, okay, into their building blocks. So their basic proteins, the basic building block of proteins is amino acids. Basic building blocks of polysaccharides are monosaccharides. And the basic building blocks of lipids, when we break down lipids, we break them down into the glycerol and the fatty acids. Remember, most lipids, or maybe I didn't discuss this, but most lipids in our diet are triglycerides. And those are the ones we've really mostly talked about. We really haven't talked about We talked about phospholipids and sterols a little bit, but triglycerides are the main ones in our diet. They get broken down into the glycerol and the 3 fatty acids. Okay, so these are all the hydrolysis products. These would be called the hydrolysis products, which are basically the building blocks of these biomolecules. Okay, so these hydrolysis products are small enough now that they can be taken up into our cells. So once they're inside the cell, all of these hydrolysis products get converted into common metabolites, okay, common metabolites. And the common metabolite inside the cell is pyruvate, okay? So monosaccharides get converted to pyruvate through a process called glycolysis, which we're going to look at in more detail. But essentially they can all be proteins, the glycerol from lipids and monosaccharides can all be converted into pyruvate. The other common metabolite is acetyl-CoA, okay, which all 3 can be converted to as well. So this acetyl-CoA ends up going through a metabolic pathway called the citric acid cycle here. And the citric acid cycle, you'll see wherever ATP is shown, ATP means ATP is being formed. So you get a little ATP formed from glycolysis. You get a little ATP formed from the citric acid cycle. But more importantly, from the citric acid cycle, we get these guys, this FADH2 and the NADH. These are sort of energy-containing nucleotides. We're going to talk about their role. But essentially, they capture electrons that are lost, okay, during the oxidation reactions here. Oxidation is loss, right? So during these oxidation reactions that occur in the citric acid cycle, electrons are lost to these guys. They get reduced, FADH2 and NADH, and they carry those electrons to a place called the electron transport chain. And the electron transport chain takes those electrons and it passes them down like an energy gradient or something more complicated than I'm even going to bother explaining. But essentially it takes those electrons and it uses them like electricity to pump hydrogens out into this special space. It uses those electrons like electricity to make this ATP. Okay? It's kind of cool actually, and I'll explain it when we get there. So that's the overview. You have these biomolecules from your food. They get broken down by hydrolysis into the basic building blocks or hydrolysis products. They then get further broken down into the common metabolites, pyruvate and acetyl-CoA. And acetyl-CoA enters the citric acid cycle. Out of the citric acid cycle, we get these nucleotides that go to the electron transport chain where the majority of ATP is actually produced. Okay, so I will come back to that overview at the end after we've looked at all of these different reactions in more depth. Okay, so before we get into these different reactions, we've got to orient ourselves to the cell again, because all these different reactions take place in different parts of the cell. And this picture is pretty detailed. It gives you a lot of the different, tells you about all the different organelles in the cell, okay? But the main one I want you to know is this one here, the mitochondria, okay? So here's your cell. It's covered with a cell membrane, and then all this jelly stuff on the inside is the cytoplasm, and there's the nucleus that contains all the DNA. And then there's these other, you know, the lysosome, Golgi, rough ER. These are all organelles you don't need to know. What I want you to know is that one of the organelles that floats around in the cytoplasm is this one here. It looks like a kidney bean with like this weird wavy like center. And it is called the mitochondria. Now in the mitochondria you have 2 membranes, right? So you have this inner one that's all folded and squiggly, and you have this outer one, which is like the outer membrane of the mitochondria. So there's 2 sort of internal spaces. This one here that's between the 2 membranes, we call this the intermembrane space, okay? The intermembrane space. It's the space between the 2 membranes. The space that's on the inside, like the middle, the inside here that is this is called the mitochondrial matrix. It's sort of like the matrix is like almost like a cytoplasm inside the mitochondria. It's like a jelly substance, okay? So that is the mitochondria. You have the outer membrane, the inner membrane, the intermembrane space, and the matrix. These folds are called cristae, but you don't need to know that. And here's telling you about the cell parts. Again, know what the cell membrane is, the cytosol or cytoplasm. They're kind of the same thing. But really, I want you to know what the mitochondria is, okay? So I guess we'll come back to that's going to become relevant when we talk about all of those different metabolic pathways and where they take place in the cell. Okay. Some more sort of introductory stuff, getting you familiar with characters we're going to see again later in this lecture, are metabolically relevant nucleotides. So nucleotides have two forms. They have a high energy form and a low energy form. And the different nucleotides that we're going to talk about that are metabolically relevant are these pairs here, the ATP and ADP. So these are showing you the high energy form and the low energy form. So ATP and ADP. NADH and NAD+, FADH2 and FAD, and acetyl-CoA and CoA, okay? 4 pairs. And here they are in picture, which I think is better to keep in mind. So this is table 12.2 from your book. So we have ATP and ADP. This stands for adenosine triphosphate because there's 3 phosphates, and adenosine diphosphate because now there's only 2 phosphates, so we've lost a phosphate group. NADH, which is the high energy form, and the low energy form is NAD+, so it has lost hydrogens, it has lost electrons because it went from a 0 charge to a plus charge. So oxidation is loss. Okay, so the oxidized form is low energy. The reduced form is high energy. Okay, and this is what it looks. You don't have to know the structure of it. I want you to know the name of it, and I want you to know which form is reduced and which form is oxidized, which form is high energy and which form is low energy. Okay. And here we go, continuing with the table. FADH2, again, the reduced form is the high energy form, and the FAD is the oxidized form. So you can see the difference in these two is up here, these hydrogens here. Okay, it highlighted them in red. So those are the 2 hydrogens that get lost, or that get gained in the reduced form, lost in the oxidized form. And then finally we have acetyl coenzyme A. And coenzyme A. So this is nicknamed acetyl-CoA and CoA, but the Co really stands for coenzyme A. Okay, so up here we get hold on, I have to think in my head. We get reduced, okay? Hydrogen is gained. Oxygen is lost and hydrogen is gained. So this is actually the reduced form here and the oxidized form here. So in this case, with acetyl-CoA, it's the oxidized form that is high energy and the reduced form that is low energy. Okay, so for for FADH2 and NADH, those are both reduced forms. They're high energy. Acetyl-CoA, it's a different story. But again, you don't have to know the structure. So if you can't tell from the name, acetyl coenzyme A versus coenzyme A, you have no idea whether it's oxidized or reduced from that. But FADH2 and FAD, you know that this one is oxidized and this one is reduced because this one has a gain of hydrogens, which is a sign of reduction, gaining hydrogens. So if you can tell from the name, I do expect you to know whether it's reduced or oxidized. So that means for NADH and FADH2, know the reduced form and oxidized form. I thought there was a consistent pattern, but apparently there's not, just for those 2. So anyway, another interesting thing to sort of point out here actually is how these structures, they point out to you the vitamins in your diet. So nicotinamide is a form of vitamin B3, niacin, And so when we say that you need vitamins, that vitamins are important in your diet, it's because you use that vitamin, that niacin, vitamin B3, to make ATP. It's a component of ATP. Same thing with vitamin B2, riboflavin is a component of this metabolically relevant nucleotide, FADH and FADH2 and FAD. And then pantothenic acid, another B vitamin, is also important here, okay, is in acetyl-CoA. So when we talk about how vitamins help us to function, and in particular if you're taking nutrition, you'll learn that the B vitamins all play important roles in metabolism. It's because they're part of these, and some of them are part of these metabolically relevant nucleotides that we can't make without the presence of those B vitamins. And we can't make the B vitamins, we have to get them from our diet. Okay, so that was sort of a tangent, side tangent. That's interesting though. So the first of these metabolically relevant nucleotides is ATP, and ATP is really what we refer to as the energy currency of our cells. So when we eat food, we talk about energy in food, we talk about it as calories. When you eat food, you get a certain number of calories from it, and those calories actually translate to how much energy you get out of the food. It has to do with how much ATP you can make from that food, which is a mixture of carbohydrates, fats, and proteins usually. So depending on how much of each it has, you can make more ATP from it. So ATP looks like this. Here's the sort of condensed structure, I suppose, in a way, just showing you the hydrolysis and condensation reactions that occur. So how ATP gives us energy. So ATP gives us energy by undergoing hydrolysis. So what happens is you have a reaction in your cell. You're trying to build a protein, okay, from amino acids, and you need ATP to help you do that. So what happens is ATP and water come in, and the ATP gets hydrolyzed. So that water molecule breaks, and it breaks off one of these phosphate groups. Okay, so you end up with a phosphate group and ADP, adenosine diphosphate, okay, and energy. So this is an exothermic reaction. It produces energy, and then that energy is captured in order to, put those two amino acids together. That energy is used to fuel work or to fuel some other reaction, or to fuel a muscle contraction. Okay, that energy can be harnessed to do other things. So the hydrolysis of ATP is actually how we get energy. It's not that like ATP is some kind of like magical like energy, like it's always drawn like a little, it's got like a star around it, and ATP has energy, but you don't really understand how it works. Well, this is how it works. It's an exothermic chemical reaction. It's a hydrolysis reaction. And that reaction actually creates energy. We've talked about exothermic reactions. So it's not so much that there's some kind of magical, energy stored in this bond right here. It's that when this reaction occurs, it's very exothermic. It releases a lot of energy. And ATP in the cell can be recycled, or I guess I should say ADP can be recycled. That when you are doing catabolic reactions, you can harness energy from the catabolic reactions in order to remake that ATP. So you take ADP here, plus an inorganic phosphate, plus some energy, and you can make ATP. So there's this sort of like recycling in the cell. We use ATP in the cell to make energy, and we use these metabolically relevant nucleotides. We use energy from those to turn ADP back into ATP through all these different metabolic pathways. Okay, so the next high energy nucleotide pairs are the NADH and FADH2 pairs, okay? These are energy transferring compounds. Okay, so not all of these energy nucleotides are created equally. They each have sort of their own function. So ATP really fuels chemical reactions throughout the cell, all kinds of chemical reactions, and mechanical reactions too. The contraction of the muscles is from ATP. So that energy can be harnessed to do work, it can be harnessed to run a chemical reaction, etc. NAD and FAD are energy transferring compounds, and that energy that they transfer usually is used to make ATP. And like I said before in the pictures, the high energy form are the reduced forms, the one with hydrogen in the name, and the low energy form is the oxidized form, the one without hydrogen in the name. Okay, and I showed you in the picture how they both actually contain a vitamin component. NADH contains niacin, vitamin B3, and FADH contains riboflavin, vitamin B2. So the last one is this acetyl coenzyme A and coenzyme A. And the energy here is released when this bond here is broken, when the thioester group is hydrolyzed, I guess. I said it was reduced, but I guess it's really hydrolyzed. And it also contains a vitamin component. So we're going to talk more about how acetyl-CoA acts as a common metabolite more so than an energy-carrying molecule. Okay, so those are all the characters. We know the different parts of the cell that are relevant. There's the cell membrane, the cytosol, and within the cytosol you've got your organelles. The most important one in metabolism is the mitochondria. And we know a little bit about how the mitochondria looks now. And we are familiar now with the different energy containing nucleotides or energy transferring nucleotides, and a little bit about the reactions that cause them to provide energy, at least for ATP. So now, how do we get those molecules? How do we create those? Namely, how do we create ATP? Well, it's a little bit of a long road. And that road starts with digestion, okay? So we eat food, here's a whole bunch of different types of food, okay? And all those foods contain these large biomolecules, the nutrients, macronutrients in your diet, carbohydrates, fats, and proteins. And when we talk about fats, we're namely talking about triglycerides, okay? So carbohydrates get broken down ultimately into glucose and maybe a couple of other monosaccharides that usually end up getting converted to glucose. We have triglycerides that get broken down into glycerol and their 3 fatty acids. And then we have proteins that get broken down into amino acids. Okay? So first let's look at how carbohydrates get digested. So a lot of the carbohydrates in our diet are these starches, like polysaccharides, like starches. And remember we talked about starches in chapter 6, I believe. There's 2 types of starch, amylose, which is the coiled chain one, and amylopectin, which is more branched. Okay, but they're both types of starch. They're both just long chains of glucose. And digestion of them actually starts in the mouth. So you have these alpha-glycosidic bonds here that get hydrolyzed. So you break water gets added, water in your saliva, okay? And also there's an enzyme in your saliva called salivary amylase or alpha amylase, which helps to speed up the chemical reaction of this hydrolysis reaction, okay? That's why if you take a cracker and you put it in water, it'll start to disintegrate some, but it doesn't get completely broken down because there's no catalyst, there's no enzyme catalyst to turn that cracker into glucose. Okay, you just get like slimy cracker. Okay, so through this enzyme catalyzed hydrolysis reaction, you end up with you get some oligosaccharides, some disaccharides, and some monosaccharides. And you keep doing this reaction until basically this whole polysaccharide has been broken down entirely into monosaccharides of glucose. Alright, so that's what this is showing here. So those oligosaccharides, so in the mouth, it's just sort of an initial, this amylase starts to break things down. So you have a combination of oligosaccharides, disaccharides, and monosaccharides in your mouth. Then the food gets swallowed, ends up in your stomach, In your stomach, you don't actually get any digestion of carbohydrates because the amylase gets denatured in the stomach because of the low pH of the stomach. And remember, enzymes only work at their optimal pH. And in your mouth, there's a pH of about 7. In your stomach, it's a pH of about 2. So that alpha amylase from the mouth, it becomes denatured and it can't function anymore. So there's actually no carbohydrate breakdown in the stomach. Then your food enters the small intestine. And in the small intestine, you get more amylase enzymes produced. Okay, so that the pancreatic amylase that is in the small intestine breaks down these oligosaccharides into disaccharides, and then maltase, lactase, and sucrase are all enzymes that break down all of the disaccharides, the 3 different disaccharides. Okay. So you end up with a whole bunch of monosaccharides here: glucose, galactose, and fructose. Mostly it's glucose though, right? So these monosaccharides now can be absorbed into your bloodstream. Okay, so this is just a little slide showing you that humans can break down amylose and amylopectin, because they have alpha-1,4 linkages, alpha-1,4 glycosidic linkages. But humans cannot break down, in our glycogen, we can break down alpha linkages, we can't break down beta linkages. Humans don't have the enzyme that breaks down these beta-1,4 linkages that you find in cellulose, which is in plant fiber. So that's why we have in our diet something called fiber, or insoluble fiber, that it's really just polysaccharides that are connected in such a way that we don't have enzymes that digest them, but certain animals do. Well, actually, I should say certain bacteria do, because like cows, for example, that graze on grass, they can break down cellulose, but it's not actually because they produce cellulase, the enzyme that breaks down cellulose. It's because they actually have special bacteria in one of their stomachs. They have 4 stomachs. That actually produce cellulase and help them break down the cellulose. So that's why cows, there's no like fiber, insoluble fiber in cows' diets. They can break it down. We can't, because we don't produce cellulase. We don't have an enzyme that can break these beta-1,4 linkages. We can only break down alpha-1,4 and alpha-1,6 linkages. So that's why fiber is fiber in our diet. That's why it doesn't get broken down. It's because we lack the enzyme to do it. So that is the skinny on carbohydrate digestion. Now for fat digestion. So fat digestion has a few more parts to it, because fats are special in that they are not soluble in the aqueous environment of our body, right? Fats, lipids are not soluble in water, and our body is 70% water. So we have some special adaptations to help us break down fats. Okay, so because dietary fats are nonpolar, we need some kind of emulsifier, some kind of chemical compound that helps us to break the fat into smaller particles so that we can then get at it more efficiently with our enzymes to break it down. So bile is a molecule that is produced by the liver and stored in the gallbladder and is released into the small intestine when you eat something fatty. And what it does is it takes your dietary lipids, which if you ever mix oil and water, the oil all like congeals together, it groups together because it's nonpolar, and it's trying to get away from the water. So it's all going to stick together in this big fat droplet, right? Same thing in your stomach, in your digestive tract. You're going to have all the fats going to coagulate together to try to escape this aqueous environment. What bile does is it acts kind of like soap. Remember how soap worked to make micelles? Those little, sort of spheres that have polar heads and non-polar tails, so all of the dirt and grease gets trapped in the middle. Same here. Bile salts form a micelle, and the fat molecules are in the middle. So you end up breaking down this giant fat globule into smaller little fat globules. And now these smaller fat droplets are easier to digest. It's easier to get enzymes in there and break them down. So what bile does is it helps emulsify. Emulsify is to break a big fat droplet down into smaller fat droplets. That's what that word means. Okay? So now that we have our fat emulsified, our enzymes can work more efficiently. So there's no fat digestion in the mouth. Well, actually there is. There's a little bit of fat digestion in the mouth and in the stomach for that matter. But here we're going to show you, it really kind of jumps into the small intestine, showing you how the enzymes, the lipases, work. So in your mouth you have lingual lipase, in your stomach you have gastric lipase, in the small intestine you have pancreatic lipase. Lipase is the enzyme that breaks down lipids, okay? And depending on where it's made, it has a new name. So they're all slightly different lipases, but they all work the same way. Lipases work to take a triglyceride, which is a glycerol molecule with 3 fatty acids, okay? And when triglycerides get broken down, they actually get broken down into, the lipase breaks them down into a monoglyceride, so a glycerol with 1 mono fatty acid, and 2 free fatty acids. This is how they get broken down. I don't know why they do it that way, but that's how it works. So you get a monoglyceride and 2 free fatty acids. Okay? And then what happens is you absorb those. You absorb these two. Now there's one. So triglyceride is too big. It's too big to be absorbed into our cells. So we have to break it down into the monoglyceride and 2 free fatty acids so we can actually absorb it into our intestinal cells. Then once it's in our intestinal cells, it actually reforms into a triglyceride. And then in order to absorb fats, so triglycerides and cholesterol, into our bloodstream, we actually have to form these specialized transfer compartments called lipoproteins. It's because it's a combination of lipids and protein. And the name of this specialized lipoprotein that we use to absorb fats from our diet into our bloodstream is called a chylomicron. It's essentially a sphere that's formed by phospholipids and proteins, kind of like a cell membrane, okay? So it's this sphere, and kind of like a micelle. And it is full of all the fats. All the fats sort of live inside of it. Okay, so now that can be absorbed into our bloodstream. It is more water soluble that way because it's like a micelle. But you can't just absorb fats straight into the bloodstream because the blood has so much water in it. It's not it's not soluble. Okay, so we have to first package them in these packages called chylomicrons, which are very much like micelles that are composed of protein and phospholipids. Okay? So that is fat digestion and absorption. The last macronutrient, proteins. Protein digestion actually begins in the stomach, not in the mouth. Hold on. I guess I feel like that makes sense to me too, because if you eat meat, and you're chewing it in your mouth, it gets all chewed up, but it doesn't really feel like it's dissolving in any way. Whereas, if you eat a cracker, it gets wet and it kind of dissolves. Or you eat a piece of chocolate that's high in fat, or, I'm trying to think of something really fatty. Butter, okay, if you just eat some butter, it would melt in your mouth, right? It feels like it's already starting to kind of break down. But you eat something that's high in protein, and it doesn't really feel like it's dissolving at all. It just feels like it's getting mashed up. So there's no digestion of protein in the mouth. It starts in the stomach by an enzyme called pepsin. And pepsin because it breaks peptide bonds. Also, you get denaturation of proteins in the stomach, which I kind of skipped over there. So before you can digest a protein, really it's important to unfold it first, right? So denaturation, that's really why you don't get any digestion in the mouth, is because proteins are too three-dimensional to really get at with any enzyme. So we have to get our proteins to our stomach first, where the pH is really low, which allows the protein to denature, and now our enzymes, pepsin, can get in there and break down the protein. So here's a question, if the stomach pH denatures proteins in our food, and pepsin is an enzyme, which means it's a protein. Why is pepsin work in the stomach? Why doesn't it get denatured? It's because pepsin's optimal pH is pH 2. It's designed to work optimally at that pH. So just because you have a very acidic environment doesn't mean all proteins get denatured, right? Some proteins are designed to function at low pHs. That's what we talked about in Chapter 10 when we talked about the optimal pH for different proteins. It's different for different proteins, but most of the proteins in your food, unless you eat a really acidic, unless it's an acidic food, most of the proteins in it are probably going to be denatured by an acidic environment, okay? So, then you move on to, so that's in the stomach, and then your food moves into the small intestine, okay, and you get more digestion of proteins by a couple of enzymes called trypsin and chymotrypsin. They both happen to break certain peptide bonds. So through all of these different stages of digestion, pepsin in the stomach, trypsin and chymotrypsin in the small intestine, you finally end up with a bunch of free amino acids, a bunch of individual amino acids, and those amino acids are small enough to be absorbed into the bloodstream. So here we go. You have a protein. It's all three-dimensional. You get it into the stomach. It gets denatured, unfolded, okay? Then it also actually starts to get digested by pepsin. That isn't shown here, but it does start to get digested. So then you move into the small intestine, and those peptides, larger peptides, get broken down into individual amino acids by chymotrypsin and trypsin. And then these individual amino acids get absorbed into the bloodstream and ultimately can go get absorbed by your different tissues. I like these little figures from your book that kind of show the chemicals in the chemical reactions sort of visually in each stage of the digestive tract. Okay, so that's how your nutrients get digested. So remember we had that step. The step one is you have these macronutrients, biomolecules in your food. They get digested into hydrolysis products. And so now we have all our hydrolysis products, our amino acids, our triglycerides, and our glucose molecules, okay? So now we're going to talk about how each of those hydrolysis products, or basic building blocks, can then be metabolized to form common metabolites, pyruvate and acetyl-CoA. Okay, so glucose, if we look at our carbohydrates, glucose gets broken down in a pathway called glycolysis. Glucose is the body's main source of fuel. It's the brain's, it's definitely the nerve tissue's main source of fuel. Like 90% of the fuel that our brain uses is from glucose. It doesn't really use fats or proteins very well to make ATP. It really relies heavily on glucose. So do your muscles. And in general, it's like the most important form of fuel. So that's why we start with glycolysis, and glycolysis is a sort of major focus. So glycolysis literally means the breakdown or lysis of glucose. So it's the pathway for catabolism of glucose. And it's a 10-step process that looks, if you write it in shorthand, it looks like this. You take glucose, one molecule of glucose, and you end up with 2 molecules of pyruvate. You should write a 2 right here in your notes, okay? 2 pyruvates. 1 glucose becomes 2 pyruvates in this 10-step process called glycolysis. So this reaction arrow here is not really referring to one reaction, it's referring to 10 reactions that occur in a stepwise fashion in order to get glucose, which is a 6-carbon molecule, into 2 pyruvate molecules, which are 3-carbon molecules. Okay? So here's my little rough diagram of what the cell looks like and where glycolysis takes place and how this happens. So you have digested your polysaccharides. You've ended up with a whole bunch of glucose that's now coursing through your blood glucose levels. Okay, and now a cell in your big toe needs to make energy. Okay, so what it's going to do is it's going to insert put this glucose transporter on the surface of its cell, and that's going to allow glucose to enter the cell into the cytosol or the cytoplasm. of the cell, and that's the first sort of step, is getting glucose into the cell. Now glycolysis can take place. This is where glycolysis takes place. It takes place in the cell cytosol, or the cell cytoplasm. Okay? So this 10-step process of glycolysis, it's a series of reactions, and in each reaction, energy gets transferred. That's what happens in chemical reactions. Electrons get transferred, energy gets transferred. So during glycolysis, it has to be this 10-step process because you want to minimize the energy transfer to each step. Because every time you transfer energy, you lose a little bit of it, and you lose it as heat. So if you did it too fast and you released a lot of heat, you would overheat yourself. So that's why there's sort of like a control mechanism. So in order to sort of minimize this heat loss, you get reactions that produce energy coupling with reactions that require energy. So same thing like redox reactions. If you're losing an electron, there's always a reaction that's taking place that's then gaining that electron. There's no sort of like loss of energy. It's always I don't know, two things that are opposite. There's always hydrolysis and condensation. There's always oxidation with reduction, okay? So typically when we're talking about energy being transferred, we're talking about it being transferred through phosphate groups, like in ATP. There's also GTP that has phosphate groups. And in this case, though, in glycolysis, it's transferred through phosphate groups being added to or subtracted from ATP or ADP in condensation or hydrolysis reactions, okay? So remember, we talked about ATP gets hydrolyzed to form ADP, and ADP can be condensed, I guess, to form ATP. So anyway, there's 10 steps to glycolysis. The first 5 steps are called the energy investing phase, because in these 5 steps, 2 of them require you to actually input ATP. It actually takes energy to break down glucose. So you're like, what? I thought you get energy from breaking down glucose. I thought that was the point of catabolism, was to make ATP. Well, you do. In the end, in the next step, you'll see that we actually make more ATP than we use, but there's actually sort of a give and take. So part of glycolysis actually requires an energy input or an energy investment. Steps 1 and step 3 both require energy investment, okay? So we use 2 ATPs here. There's a star at this step because we're going to come back to it and talk about that step of the pathway and why it's very important in regulating glycolysis. Then the next 5 steps, the second half of glycolysis, you actually make ATP. You generate 2 ATPs at step 7, and you generate 2 ATPs at step 10. So overall, in glycolysis, you use 2 ATPs, but you make 4 ATPs, so there's sort of a net gain of 2 ATPs at the end. Do I have that on this slide? No. But so that's how glycolysis makes ATP. So at the end of glycolysis, you've actually generated a net of 2 ATPs. You've also generated a high-energy nucleotide, 2 high-energy NADH molecules here at step 6. There's a reduction reaction that goes on, or a redox reaction. So you actually generate 2 NADH molecules and 2 ATP molecules as a result of glycolysis. So the book also walks you through each of these reactions step by step, and I'm going to do it to some extent. Just parts that are particularly interesting that I think. Okay, so this first step I just want to point out is, so you have glucose molecule here, and what happens is, in the first step, hexokinase, which should be familiar to you because it was a big focus of Chapter 10. Chapter 10 chose to use the molecule, the enzyme hexokinase, as the example enzyme and how it binds to glucose and ATP and it adds this phosphate group to the glucose molecule. Now the reason for that is because when you take glucose up into the cell through that glucose transporter, it could just as easily go back out the other side. So in order to keep it in the cell, once you take it in the cell, hexokinase adds this phosphate group to it, and now it won't fit back through that channel. So now it's stuck in the cell. So that's the first reaction of glycolysis. So that one I wanted to point out because hexokinase is an enzyme we've seen before. So I expect you to, and that we've looked at in detail how the mechanism of how it works, how it binds glucose and ATP and causes the transfer of that phosphate group. Okay, so that's a step that does require the energy input, ATP input. And step 2, essentially what happens in the next step is you get another phosphate. It turns from a glucose ring into a fructose ring. So it isomerizes. So there's a shift in the shape of the ring. Then we add another phosphate group in step 3. This is an important step, okay? This step is catabolized by an enzyme called it's catalyzed by an enzyme called phosphofructokinase. It's a mouthful, phosphofructokinase. Also, in like the biochemistry world, people just call it PFK for phosphofructokinase. Okay, phosphofructokinase, it adds another phosphate group, but the reason that it uses ATP to add another phosphate group to this molecule that we originally was a glucose. And this happens to be the regulatory step of glycolysis. So if cells have enough glucose, they can basically put a halt on phosphofructokinase and say, let's stop glycolysis. We have enough energy. We don't need to break down any more glucose. You can stop breaking down glucose. This phosphofructokinase is the one that is sort of regulated, okay, but we'll come back to that. So then the next step is that this molecule here gets broken into 2 and gets converted. There's a few more steps. I'm not going to walk you through all of these, but I don't really want to focus on any of these. As we've already talked, I want you to know which steps use energy. So steps 1 uses ATP, step 3 uses ATP. I want you to know hexokinase is the enzyme in step 1, and I want you to know that phosphofructokinase is the enzyme in step 3, and that step 3 is the regulatory step. You don't really need to know about these others, but it's kind of interesting if you look through it, because you can kind of see it's like one thing happening at a time. So this gets cut into 2. These 2 molecules get converted into, they isomerize, so they get converted into a different isomer that happens to be this molecule here. And then that molecule gets phosphorylated, right? No, it's already phosphorylated. That molecule then gets converted into another molecule. It gets phosphorylated. And in that phosphorylation step, that energy is collected or gathered captured by this high energy nucleotide NAD, which gets converted into NADH, which is high energy, the high energy form. It gets reduced. And so I would like you to know that reduction step occurs at step 6. And then at step 7, we actually produce ATP. We technically produce 2 ATPs because technically we have 2 of these 3 carbon molecules as a result. So this, kind of goes, you should think of it as having 2 branch points. So now all the steps after step 5, everything is sort of, there's 2 of them, right? Because we have broken this into 2 3 carbon molecules. So now we have 2 reactions 5 through 10 all happen in doubles, in pairs. Okay, so we have this reaction actually happens twice. There's 2 of them. So we get 2 NADHs. We get 2 ATPs at step 7. We get 2 ATPs again at step 10. Okay, the last step here. And the final product of glycolysis after reaction 10 is 2 molecules of pyruvate, because remember there's 2 parallel pathways going on here. So you end up with 2 molecules of pyruvate. So again, this is just showing you the net reaction of what happens in those 10 steps. Think this reaction arrow is actually all 10 steps of glycolysis. So this is a good sort of thing to know, this reaction here. 1 molecule of D-glucose plus 2 NAD plus the low energy molecules plus 2 ADP, low energy molecules, plus 2 phosphates, which then get added on to those ADPs to make ATP. So you end up with 2 pyruvates, 2 NADHs, and 2 ATPs, and 2 free hydrogens. But we don't worry about the hydrogens or the inorganic phosphates. What I want you to know really is that glucose becomes 2 pyruvates, 2 NADHs, and 2 ATPs. This is the sort of net output of one glycolysis 10-step reaction. Okay? So glycolysis is a metabolic pathway of 10 steps to get from glucose to 2 pyruvates, 2 NADHs, and 2 ATPs. So these ATPs, bam, that's instant energy right there. That's the ultimate result of all of these metabolic pathways we're talking about is to make ATP. So after glycolysis, we've already made 2 molecules of ATP. Great. We can use those right away for energy. These NADH molecules we're going to see again down the road. And now we're going to see what happens to these 2 pyruvate molecules to continue to, because we're going to continue to break this down to make more ATPs as well. Okay? So, before I show you what happens to that pyruvate, we'll come back to it in a second, I want to go back to talking about the regulation of glycolysis. So remember I said glycolysis is regulated at that 3rd step where phosphofructokinase is. Okay? So this is really true of pretty much all metabolic pathways. They all have some point, some step that's regulatory. That is, they all have some place in the reaction where, depending on what the needs of the cell are, the cell has the ability to turn that pathway on or off. And in the case of glycolysis, if there's a lot of ATP in the cell, well, then we don't need to keep breaking down glucose for energy because we can't store that much ATP. So therefore, if there's lots of ATP, we don't need to keep breaking down glucose. So what happens is, here's a close-up of step 3. So you have fructose 6-phosphate is what molecule exists at step 3. And ATP is added. Remember, step 3 is one of the steps where ATP is used. And you form ADP in this fructose 1,6 bisphosphate. Okay, this is the step 3 reaction. And it's not really important. You don't need to know the details of this reaction. You just need to know that ATP is added and that phosphofructokinase is the enzyme that catalyzes it. So anyway, phosphofructokinase, its activity as an enzyme is inhibited by ATP. So if there's high levels of ATP in the cell, it actually inhibits phosphofructokinase and puts a halt on glycolysis. So it's kind of genius. If the levels of ATP in the cell are low, then you have less inhibitor around and the phosphofructokinase works more efficiently. So the more ATP there is, it kind of sort of acts as an inhibitor to phosphofructokinase. So this is called a negative feedback loop. Okay, it's the same way that your thermostat works in your house. So you set it to a temperature like 70 degrees, and when the heat builds up in your house, it actually tells the thermostat to turn off. It says, it's warm enough in here, stop making heat. And then as it cools down, the thermostat kicks in and it starts producing heat again. Okay, that is negative feedback. It means that when you produce a lot of the product, that product feeds back to negatively inhibit the pathway, right? Negative feedback. The product, lots of product actually inhibits the pathway. So that's how glycolysis is regulated. It's regulated by negative feedback. The more ATP there is in the cell, the less glycolysis you have going on. Okay? So this is kind of an interesting tidbit that your book goes into, which I'm glad about. So remember, there's 3 different monosaccharides as a result of carbohydrate breakdown. Glucose is the major one. Then you also have galactose and fructose. So cells actually, I think with galactose, they just convert it to glucose. I'm not sure if we have a separate pathway to break down galactose. However, we do have a separate pathway to break down fructose. We can do both. We can either convert fructose to glucose, or we can break down fructose on its own. So the fructose breakdown, all right, so here's looking at fructose metabolism versus glucose metabolism, okay? So it's similar. In this first step, both of them have a first step that uses ATP and phosphorylates the sugar so that it stays inside the cell. Then what happens to fructose is it gets broken down into 2 3-carbon molecules, kind of like what happens here at step 4 of glycolysis, okay? So essentially what happens is these 2 products here of fructose metabolism, glyceraldehyde 3-phosphate and dihydroacetone phosphate, check it out, dihydroacetone phosphate, glyceraldehyde 3-phosphate. These are the same products as at step 4 of glycolysis, okay? So these fructose breakdown products, G3P is what we call this for short, and I think we call it DP, DAP maybe, I can't remember what it's called and what I used to call it in biochemistry. But anyway, these 2 products then can feed into the glycolysis pathway, so you can draw an arrow here that these products of fructose breakdown then feed into glycolysis and just follow the pathway and make ATP the same way that you do with glucose. But the difference here is that glycolysis is regulated at this step right here, step 3. If there's a lot of ATP in the cell, we stop breaking down glucose. But fructose bypasses this step. So we get these products here that feed into glycolysis. So even if the cell says we have too much ATP, stop breaking down glucose, it'll still break down fructose, okay? So what happens is fructose bypasses this regulatory step, and it produces extra pyruvate, so extra breakdown products. And that pyruvate then gets converted to acetyl-CoA, and acetyl-CoA is what gets stored as fat. If you stop glycolysis here and you have excess glucose, you can store that glucose as glycogen, or I think you can even get rid of it in the cell, like out like through the blood. But usually you would store it as glycogen, whereas fructose, if you break down extra fructose, you get extra acetyl-CoA, which gets stored as fat. So that's part of the argument. This chemistry here is part of the argument about why high fructose corn syrup is making us fat, because fructose breakdown isn't regulated by cells, and so it can result in excess acetyl-CoA and excess fat storage. Interesting, right? Okay, so now let's get back to pyruvate. Pyruvate is the end product of glycolysis. Remember, we have that 6-carbon glucose molecule, we go through the 10 steps of glycolysis, and we end up with 2 pyruvate molecules, okay? So there's 2 different fates that pyruvate can have, and those 2 fates depend on the presence or absence of oxygen. So if oxygen is around, if there's oxygen around, and usually there is in our bodies because we breathe in oxygen, our cells need oxygen for this purpose, really, for cellular respiration, so there's usually oxygen around. Usually pyruvate goes down this pathway, the aerobic pathway. Aerobic means pertaining to air or oxygen, okay? So if that happens, then pyruvate gets oxidized to acetyl-CoA. It gets oxidized to acetyl-CoA. We undergo aerobic metabolism and we produce a ton of ATP, 36 to 38 ATPs produced for 2 pyruvate molecules. Now, if there isn't any oxygen around, for instance, if you are exercising and in the muscle tissue, it's working so hard that it's spending ATP so quickly, and using oxygen so quickly that it actually becomes like there's very low oxygen there because it's using it quicker than your body can get oxygen to your muscles. Then you get anaerobic conditions, okay? And in anaerobic conditions, without oxygen, pyruvate gets reduced. So it makes sense. And when there's lots of oxygen around, pyruvate gets oxidized. If there's not a lot of oxygen around, then pyruvate gets reduced to a molecule called lactate, which gets converted to lactic acid. And we can actually make some energy from lactic acid. So again, here's just showing you sort of the chemical reactions. Under aerobic conditions with lots of oxygen around, okay, you have pyruvate and you have coenzyme A and NADH or NAD+. And you get, so coenzyme A comes in and combines with pyruvate essentially to form acetyl coenzyme A. Acetyl coenzyme A. And you get reduction of NADH. So you actually produce an NADH there. So that's why you don't need to know the structures. You don't need to know this reaction. What I want you to know is that under aerobic conditions, pyruvate gets oxidized, and the sort of the result of that oxidation reaction is acetyl coenzyme A and NADH. Okay, those are the major products I want you to know. Acetyl coenzyme A and NADH are formed. Under anaerobic conditions, without oxygen, pyruvate gets reduced, so hydrogens are gained here, into lactate. Hydrogens are reduced into lactate, and you actually use NADH in that reaction. So, this is just showing you where this actually occurs. So remember we said glycolysis in the cell occurs in the cytoplasm. Glucose gets taken into the cell from the bloodstream, and then glucose undergoes glycolysis to make pyruvate. We're all still in the cytosol or the cytoplasm. Then pyruvate can go one of two ways. If there's no oxygen around, if it's anaerobic conditions, the pyruvate gets converted to lactate. And if there is oxygen around, then the pyruvate gets converted to acetyl-CoA, which you should write here, acetyl-CoA, which then gets actually sent to the TCA cycle, the citric acid cycle. That's going to be the next metabolic pathway on our list. So glycolysis doesn't actually require oxygen to be around. You can undergo glycolysis with or without oxygen. It's that after glycolysis and you have pyruvate, what happens to pyruvate depends on whether there's oxygen around. So if there is not, you form lactate. If there is oxygen around, you form acetyl-CoA. And acetyl-CoA then gets moved into the mitochondria and into the matrix of the mitochondria where the TCA cycle occurs. Okay? We're going to talk about the TCA cycle in a minute in more detail. But after the TCA cycle, you get a bunch of NADH and FADH2 that are carrying electrons. They are reduced, so reduction is gain of electrons. So they carry those electrons to what we call the electron transport chain, where a reaction called oxidative phosphorylation occurs, and that's how we make ATP. So this is sort of, I like this picture because it's a nice overview of where all these things happen in the cell. Glycolysis in the cytoplasm, then the citric acid cycle in the matrix of the mitochondria, and oxidative phosphorylation also occurs in the mitochondria, and we'll talk about more specifically as we get there. So now we're talking about the citric acid cycle. So the citric acid cycle, for some reason, has multiple names. It's also called the Krebs cycle because it was first identified and characterized by two scientists, a married couple with the last name of Krebs. So the Krebs cycle. And then also it's called the tricarboxylic acid cycle, or the TCA cycle. So in different textbooks, different things, you'll see it called TCA cycle, the Krebs cycle, the citric acid cycle. All the same thing. I might have figures in the book that, or in the lecture that call it, like for instance, the last slide called it the TCA cycle. Your textbook calls it the citric acid cycle. So they're all the same thing. So the citric acid cycle is a series of 8 reactions. So glycolysis was 10 reactions. Citric acid cycle is 8 reactions. That's what when we say a metabolic pathway, It's a metabolic pathway that consists of 8 reactions. And it takes that citric acid, or sorry, it takes the acetyl-CoA molecule, that common metabolite, acetyl-CoA, and it uses it to make undergoes chemical reactions, and the output of the citric acid cycle is carbon dioxide, NADH, and FADH2. These 2 molecules here being the important energy-carrying nucleotides that take the electrons that are captured in this metabolic pathway and then transport it to this last pathway that is involved in making ATP. So the carbon dioxide that's produced is part of our body's waste, our metabolic waste. That carbon dioxide then gets expelled goes back into the bloodstream and gets expelled by the lungs, right? Okay, so here is the figure from your book going through the TCA cycle. You do not need to know any of the steps of the TCA cycle or the citric acid cycle, whatever. What I really want you to know is what the sort of overall products are from the TCA cycle. I want you to know that we get NADH, FADH2, and actually this GTP here is as good as an ATP. It's very similar. So you actually do produce 1 ATP, and it looks like 1, 2, 3 NADHs and 1 FADH2 for each round of the citric acid cycle that you go through. How many acetyl-CoA molecules do you get from 1 round of glycolysis? You get 2 acetyl-CoA molecules, because you get 2 pyruvates from 1 round of glycolysis. Each of those pyruvates gets converted into an acetyl-CoA under aerobic conditions. So for 1 glucose molecule, you actually get 2 turns of the citric acid cycle. Okay? So here I have another figure here where it actually calls it the Krebs cycle. So you can see how it really does vary between different textbooks what they call it. So again, this TCA cycle, the Krebs cycle, the citric acid cycle, whatever you want to call it, takes place in the mitochondrial matrix. So acetyl-CoA gets transferred from the cytoplasm into the mitochondrial matrix so that it can undergo the Krebs cycle inside the mitochondria. Okay? So here's my thing about how many turns of the citric acid cycle. Okay? So 1 turn of the citric acid cycle produces this: 3 NADHs, 1 FADH2, and a GTP, which is kind of the same thing as ATP. We can convert it into ATP. Also, these are all like sort of forms of energy that we can use. You also get 2 carbon dioxide molecules, which is a waste product, and a coenzyme A, which can then go back and become acetyl-CoA once it combines with a pyruvate. Okay, so here I have underlined, I just have it written down in sort of reaction form. I've underlined here for you the high energy or the energy molecules that are produced from 1 turn of the citric acid cycle. But remember that 1 glucose molecule makes 2 pyruvates, which make 2 acetyl-CoAs, so there are actually 2 turns of the citric acid cycle for each molecule of glucose. So this slide has sort of a compilation for you, showing you what forms of energy are formed during each of the steps we've talked about. So you have a molecule of glucose. First thing that happens, the 10-step process of glycolysis. And the end result of glycolysis is you get 2 NADHs and 2 ATPs. Okay? Then you have the oxidation, the 1-step oxidation of pyruvate into acetyl-CoA. There's technically 2 pyruvates, so you get 2 acetyl-CoAs, and you also get 2 NADHs from that, those 2 reactions. So oxidation of 2 pyruvates also results in 2 NADHs. Then you have the citric acid cycle, where you do it twice, remember, because you have 2 acetyl-CoAs from 1 glucose molecule. So you end up with a total of 6 NADHs, 2 FADH2s, and 2 ATPs. But what I'm pointing out here is, didn't we say that the whole point of breaking down glucose is to get ATP, that we use glucose to make ATP? So where's all the ATP? We've made 4 ATPs total so far, okay? The rest of the ATP is going to come in the last metabolic pathway in this chain of events, which is called the electron transport chain, okay? And that's where all these NADHs and FADH2s come into play. They're all electron carriers. They're reduced. Oxidation is loss, reduction is gain. These are the reduced forms that have gained electrons, and they carry those electrons off to the next and last stop in the pathway, which is the electron transport chain. And we use those electrons kind of like electricity to pump this sort of machine that makes ATP. All right? So these reduced electron carriers, NADH and FADH2, deliver electrons to the electron transport chain. The electrons journey through the inner membrane of the mitochondria, resulting in proton pumping into the intermembrane space. So what does that mean? So you have this electron transport chain, which is a series of proteins that are in, so remember you have in the mitochondria, you have the outer membrane, you have that inner membrane, which is folded, and then between them you have a space called the intermembrane space, okay? So what happens, and then here's the electron transport chain. It's a bunch of proteins that are embedded in the inner membrane, okay, of the mitochondria. And you get NADH and FADH2 come in, and they drop off their electrons at different points to different molecules here in the electron transport chain, and those electrons get transported through these proteins. So essentially they just get passed, the electrons get passed down this, they get passed from complex 1 to complex 2 to complex 3 to complex 4. And the passing of those electrons is sort of like a flow of electricity. And that electricity or energy is used to have these complexes pump protons from the matrix, from the inside of the mitochondria, into this intermembrane space. Okay, so you see all these plus charges building up here in the intermembrane space, and there's only like one plus charge here because they're all getting pumped out. Okay, this is a form of active transport. We are using energy from these electrons in this electron transport chain to pump protons into this space. Well, what happens is it creates sort of a pressure. It creates what we call a proton gradient. There's a lot concentrated in the intermembrane space, and there's not a lot on the inside. And what happens is, cells don't really like, things like to be balanced in biology. Cells don't like having this imbalance. It creates a pressure here. So you kind of have these protons in the intermembrane space are like pushing, wanting to go back in the other direction. It's like actually like a physical force. And if you let them go back in, and this molecule here, ATP synthase, which is the last, which is really complex 5 of the electron transport chain, allows those protons to come back into the other side. And when it does that, it's using that force, that pushing force, in order to get energy from that. It uses that force to actually propel. It's got like this little propeller, like a piston, or it's not a piston, I don't know, like a turbine, like how wind machines work, okay? And so it uses the protons going in, power this little propeller that then uses that energy to make ATP. Probably in a very similar fashion that windmills make energy from the wind, but I don't really know much about that, so, but I imagine it's similar. Okay, so let me just back this up a little bit. No, not to here. To here. So the electron transport chain, what it is technically is it's 5 membrane-bound complexes, so basically 5 proteins that are embedded in the membrane, C1, C2, C3, C4, complex 1, 2, 3, 4, and 5. Complex 5 is also called ATP synthase. You can see that here in this picture. I think I have it written in another slide. There's also those are the proteins that are embedded in the membrane, and that means they're stuck there. There's also 2 mobile carriers. So because those proteins are like embedded in the membrane, they can't move. But there's two electron carriers, coenzyme Q, I spelled this wrong, coenzyme, there's no N there, coenzyme Q, and cytochrome C. And you can see them here in this picture, this little orange ball that says Q and a little orange ball that says C here. So coenzyme Q is mobile. It lives inside this inner membrane, okay, and it can transfer electrons from C2 to C3. And cytochrome C also lives in this inner membrane, and it can carry electrons from C3 to C4. Okay, so you have 5 complexes that are embedded in the membrane, and then you have 2 that are carriers, that are mobile carriers. And the point, again, is to take these electrons and pass them down in this chain. So here's a better picture. This one's from your book. And so complex 1, 2, 3, 4, and 5. Remember, complex 5 also can be called ATP synthase because this is the one that synthesizes ATP. Okay? So you have NADH and FADH2. And NADH, it'll come and it'll drop its electrons off here at complex 1. FADH drops its electrons off here at complex 2. Okay? So which one do you think, and remember, so the electrons fuel this pumping of protons up into the inner membrane space, and the more protons that are up there, the more energy there is to make ATP. So, which one do you think provides more, or makes more ATP, NADH or FADH2? And I should also point out, NADH or FADH2, which one do you think makes more energy? I'm looking for my slide that shows it. Okay, here it is. So hopefully you said NADH. NADH makes more energy. Oh, I have it. I'm kind of going out of order. Why do you think NADH produces more ATP than FADH2? It's because, look at where they drop off. Okay? NADH drops off at complex 1. So its electrons actually get to travel further. They pump more protons out. So for 1 molecule of NADH, you make 2.5 molecules of ATP. For 1 molecule of FADH2, you make 1.5 molecules of ATP because it drops it off later in the chain, so you don't get as many protons pumped. So there you go. Okay, so this is what happens. These electrons get dropped off, and complex 1, complex 3, and complex 4 can all pump protons out of the cell. Okay, I don't care. You don't need to know which complexes pump protons or how many protons they pump. I just want you to know that some of the complexes have the ability to pump protons out of the matrix and into the intermembrane space of the mitochondria. So that space between the two membranes gets full of positively charged protons. And what you have here is you have this energy, you have a charge gradient. So it becomes more negatively charged in the matrix, more positively charged in the intermembrane space. And those protons are attracted to that. They want to come back in. They don't like being concentrated all together in this intermembrane space. And so they start pushing. They want to it's like a mob wanting to come back inside to the matrix where they have more room to spread out and more negative charges to balance them. Okay, so they're not allowed back in anywhere. The membrane is sealed to protons that won't let them through. The only way they can get through is here at complex 5, also known as ATP synthase. And complex 5 is like a proton channel, and it can open up and allow protons to come through. And when they come through, they come rushing through with force because they're so excited to get back into the matrix that rushing and that pushing actually provides energy for the production of ATP. So ADP plus phosphate becomes ATP, and that energy is harnessed from this proton pumping. Also, so that's the phosphorylation of ATP occurs here, or phosphorylation of ADP, meaning adding phosphate to ADP to make ATP. So that's this word here. When we talk about electron transport chain, we're talking about a chemical process or a chemical reaction called oxidative phosphorylation. And it's called that because ADP gets phosphorylated to make ATP, And that reaction is actually also coupled with this oxidation reaction here. So when those protons come back in the cell, they combine with oxygen to form water. So that reaction actually releases a lot of heat. So I guess that's more what it is. It's not so much like I was trying to like simplify it and make it like a simple analogy of all those hydrogens pushing in, okay? Really what it is when those hydrogens come back in the cell, two things happen, okay? You get two reactions. One is those hydrogens combine with oxygen, they get oxidized, okay, oxidative, to form water. Water is actually a byproduct of metabolism. And that is actually a very exothermic reaction. So the energy from that reaction is harnessed here in an endothermic reaction. So it takes energy to phosphorylate ADP and make ATP. But there's a lot of energy lost in this reaction that gets gained by this reaction. So oxidative phosphorylation is a way of coupling a very exothermic reaction, one that gives off a lot of energy, to a very endothermic reaction, one that needs a lot of energy, okay? So the chemical energy from here is used to make ATP, and that ATP can now be used elsewhere in the cell to fuel reactions, okay, as energy for reactions. So that is oxidative phosphorylation. It occurs as a result of these high energy electron carriers dropping off their electrons at the electron transport chain, pumping protons into the intermembrane space, and then when those protons come back in, we oxidize them. That energy from that reaction then is used to phosphorylate ADP, and finally we get ATP. So, this is just a slide explaining everything that I just explained, that you have your high energy nucleotides, NADH and FADH2, they drop off their electrons. That's step one. Step two is electrons flow through those complexes and fuel the pumping of those protons out into the intermembrane space, okay? This builds what we call an electrochemical gradient. There's more positively charged protons in the intermembrane space than there are in the matrix. That's called a gradient when you have an uneven distribution, okay? So then protons want to come back in, and when they do come back into the matrix, they do it through the last protein complex, complex 5, which is also called ATP synthase, and that coming back through provides the energy that's necessary for ATP synthesis, okay? And like I said, ATP synthesis, I just explained everything on one slide, and I didn't realize I had other slides to explain it all. But so I'm repeating myself, but that's good because it gives you a second chance at the material, okay? So ATP synthesis, remember, involves a coupling, okay? Coupling, joining of two reactions, in order to undergo oxidative phosphorylation. So you have all these protons get pumped out into the intermembrane space, and then the protons come back in, the protons come back in here at ATP synthase. This is the complex 5, okay? And they come back in here and they get oxidized to make water, and then you get ADP. Oh, no, sorry. This is showing you uncoupling. So I already showed you coupling, what reaction coupling was here. Okay, the protons come back in, they get oxidized, that's exothermic, and then that energy is used to make ATP. Okay, so what happens is, if you don't have that proton gradient, okay, you can't make ATP. That coupling is very important for the production of ATP. So if you somehow disrupt the membrane so that protons can freely move in, okay, so this is what this is a slide of. So it's showing you this. Let's say this is complexes 1 through 4. They pump protons out, okay. But this molecule here is a channel that allows the protons to just freely flow back in. They can just flow back into the matrix. They don't have to go through ATP synthase. So if they don't flow back in through ATP synthase, you can't make ATP. Okay, but what happens is these protons still get oxidized, okay, and that's a very exothermic reaction. We said that that's a very exothermic reaction. If that energy is not getting used, coupled with this reaction here, and used to make ATP, it's actually just getting released as heat. So when you uncouple the electron transport chain, you actually get a lot of heat produced. So if you disrupt the inner membrane somehow, you uncouple these reactions and it causes heat production as opposed to energy, ATP production. So what's interesting is this is how you actually make energy in some specialized cells in your body. Well, adults don't really have very many at all, but babies have quite a bit. And also, like bears, like polar bears and animals that hibernate or live in very cold environments also have a lot of this tissue called brown fat, okay? And brown fat, in brown fat cells, they produce these proteins that uncouple the electron transport chain. And what that means is that those cells actually produce a lot of heat. Now, babies don't have, their body's thermostat doesn't work very well. Like they haven't quite matured. Their biochemistry isn't such that they can regulate their body temperature like we do by sweating and by doing other things. And so it's important for them to stay warm. And so their bodies produce heat through thermogenesis. They have a lot of brown fat. And as you get older, you start losing your brown fat and it becomes yellow fat, regular fat cells. So, and the way that it produces that heat is through this uncoupling, what we call thermogenesis, generation of heat. Okay? So it's kind of cool. It's kind of a way to produce heat as opposed to ATP. So another thing that can produce, so what happens is if your cells have a lot of ATP, what happens? If there's a lot of ATP, your cells tell your body to stop breaking down glucose and to store it somewhere, to store it as fat or to store it as glycogen. And any extra acetyl-CoA can get stored as, if you make a lot of acetyl-CoA and you turn that into ATP, if you have extra acetyl-CoA, that can be stored as fat. So if you have a lot of ATP, it can act in that negative feedback loop and tell you to start storing any extra energy molecules you have. Well, there's a product on the market that used to be on the market. I don't know if it's still legal or not. I guess it is. Called DNP, which the chemical name for it is 2,4-dinitrophenol, and it's called DNP. And what it is it's an uncoupler, okay? And it was widely used in the 1930s as a diet aid. And what happens is it uncouples this. So DNP acts like this guy right here. It uncouples this reaction so you don't make lots of ATP. I mean, you make some, but you don't make ATP as efficiently. So then your body has to break down more carbohydrates, more fats. But what also happens is you make more heat. So it can be dangerous. People who use it can result in fatal hyperthermia. So their body temperature rises and they essentially die from a high fever. Okay, so it can be dangerous because it uncouples this reaction. And the positive side of it for those who use it as a diet aid is that you don't produce as much ATP, so therefore you continue to break down excess glucose and fatty acids to make more ATP. The problem is that all those glucose and fatty acids you're breaking down, instead of generating ATP to fuel your body's reactions, it generates heat, which raises your body temperature, which can be very dangerous. Okay? So that's what this says. The body can't make ATP as efficiently and thus burns more glucose and fat to make up for the inefficiency, but it can result in dangerous hyperthermia, so I don't recommend its use. But it's an example, a real-world example of how something works using this because it uncouples that reaction. Okay? So I already sort of told you about this. So now we're talking about numbers of ATP production, how much ATP is actually produced in the complete oxidation of one glucose molecule. Okay, so one glucose molecule has to go through all of these pathways. All right, so that's why it's a little confusing, because we still only are talking about glucose here. We've only talked about glucose, which is, again, our major source of fuel. So that's why we're focusing on it. So glucose goes through glycolysis, then it goes through the citric acid cycle, and then it goes to the electron transport chain. Those are like the three major things that happen. If you want to get a little more nitpicky, we've got glycolysis, which takes glucose and produces two pyruvates. And the sort of energy yield from that, you get the reduced nucleotides and the numbers of ATPs from each of these reactions. So one glucose molecule results in two NADHs and two ATPs. Those two NADHs, when you take them to the electron transport chain, will produce 5 ATPs, is what it's saying. Okay. Then the next thing is the pyruvates that you have after glycolysis get converted to acetyl-CoA. That produces two NADHs, and when you take those to the electron transport chain, they end up producing 5 ATPs. Then from the citric acid cycle, we have 6 NADHs and 2 FADHs formed, which if you deliver those to the electron transport chain, you end up with 15 and 3 ATPs. Also, as part of the citric acid cycle, you produce 2 ATPs, technically 2 GTPs that get converted to 2 ATPs. So all in all, the grand total from one molecule of glucose is 32 ATPs, okay? And this is just showing you the breakdown of where they all come from. So that's pretty cool. 32 molecules of ATP from one molecule of glucose. That's why it's such a great fuel source. So we spent most of the chapter talking about glucose as a fuel source, but we can also use fats and proteins as a fuel source, and that's what the last part of the chapter kind of deals with. So any excess glucose we have can be stored, as you know, as glycogen in our bodies. It's a polysaccharide, a branching polysaccharide. And it's happens to be stored mostly in the liver and the muscles is where we store most of our glycogen. And glycogen, we store enough to fuel our body's activities for a couple of hours. So not like if you're stranded on an island, you're not going to get by based on your glucose stores. Your glucose stores are basically there to get you through between meals. So the glycogen that's stored in your liver is there. So like if it's been a while since you've eaten and your blood sugar starts to drop, it starts breaking down glycogen in the liver and releasing that into the blood. Glycogen in your muscles is there to fuel exercise. So if you decide to go for a jog and you suddenly your body suddenly starts using more glucose than it was using 5 minutes ago when you were sitting on the couch or tying your shoes, that there's energy stored that can be quickly broken down and used to fuel that jogging activity. Okay, glycogenolysis is the process of breaking down glycogen to use that glycogen as energy. So, and then another word here, gluconeogenesis, is the formation of glucose from other non-carbohydrate molecules. So, for instance, amino acids some amino acids can actually be converted into glucose by this process of gluconeogenesis. So, glucose, important fuel source. We store some of it in the form of glycogen. We can break down glycogen by glycogenolysis, and we can make glucose from other molecules. If we really get desperate, we can actually generate glucose from like amino acids. But we can also use fats and proteins for energy. So fats, the digestion of fats, well, we talked about the digestion of fats, of breaking down the triglycerides into glycerol and fatty acids. And fatty acids can be, so like sort of the glycolysis breakdown pathway of fatty acids is called beta oxidation. So in glycolysis, glucose gets oxidized to acetyl-CoA. In beta oxidation, fatty acids get oxidized into acetyl-CoA. Okay, so you have a long fatty acid here. Okay, and it gets broken down basically 2 carbons at a time down here, 2 carbons at a time to form acetyl-CoA. So what happens is CoA binds to the end of the fatty acid. It's a 4-step process that produces some FADH2 and some NADH. And in the end, those last two carbons of the fatty acid break off, and you end up with a shorter fatty acid. So this was initially an 18-carbon fatty acid, and what the result of it is a 16-carbon fatty acid and an acetyl-CoA. And then you repeat the procedure again until you have broken this whole chain down into acetyl-CoAs. So, quiz question here. This fatty acid, 18 carbons, how many acetyl-CoAs can you get from this fatty acid? The answer is 9. You get each of these two carbon groups break off to form acetyl-CoA. So an 18-carbon fatty acid chain can form 9 acetyl-CoAs. Okay, 2 carbons at a time. So it's kind of a long, annoying procedure to break down a fatty acid, because even though you get a lot of acetyl-CoA, one glucose molecule produces how many acetyl-CoAs? 2, right? One glucose becomes 2 pyruvates, which becomes 2 acetyl-CoAs. One 18-carbon fatty acid becomes 9 acetyl-CoAs, which actually can produce a lot more ATP than one molecule of glucose. But the problem is this beta oxidation pathway is a pain in the ass, because you have to do it one step at a time. So you do this 4-step pathway to result in one acetyl-CoA, then you have to go through and do it again and again and again, and it's a little bit slower and less efficient than glycolysis is, which is why glycolysis is the preferred sort of method. So let's look at how many, let's compare here. 1 round of glycolysis versus 1 round of beta oxidation. How much energy do you get? Okay, so glycolysis produces 4 NADHs, beta oxidation only produces 1 NADH. Glycolysis produces 2 ATPs. Beta oxidation doesn't produce any ATPs, but it does produce another FADH2. So maybe I should have put this up here with the other energy-carrying nucleotide. And then we get 2 acetyl-CoAs from glycolysis and 1 acetyl-CoA from beta oxidation. So that's why glucose is a more efficient source, because 1 round of glycolysis produces more than 1 round of beta oxidation. Now, one fatty acid can undergo a bunch of rounds of beta oxidation. So this 18-carbon chain here can undergo 9 rounds, or 8 rounds, I guess, of beta oxidation, which is going to produce 9 acetyl-CoAs, which ends up producing 90 ATPs, okay, from those acetyl-CoAs. Plus, you get the NADHs and FADH2s that are produced by each round of beta oxidation. You add how much ATP that's going to produce in the electron transport chain. When you drop these off the electron transport chain, you can get from one 18-carbon fatty acid, you actually get 122 molecules of ATP compared to 32 molecules of ATP from glucose. So that's why we use fat as our major form of energy storage. We can't break it down as efficiently. It's not as efficient because 1 round of beta oxidation doesn't produce as much energy as glycolysis. But we can undergo several rounds of beta oxidation. It takes a little longer, but we can actually get a ton of energy from fat. So that's why fat is really an important energy source in terms of long-term energy storage, and also in terms of producing energy when we're like at rest or doing things that we don't need energy very quickly, that our bodies have a little more time to break down those fat molecules. So your fat stores are the ones that'll keep you alive for several days or possibly weeks if you're on like stranded on an island somewhere. Not your carbohydrate stores. It's your fat stores that will keep your cells energized with tons of ATP. Okay? So, what happens if you get too much fat breakdown and therefore too much acetyl-CoA production? So, in the absence of carbohydrates and you get a lot of fat breakdown, you get a lot of acetyl-CoA buildup. Okay? So, when that happens, that's not a good thing. Because remember, glycolysis is if you have a lot of carbohydrates around and you're breaking down carbohydrates, you can regulate that pathway. When you start, if you don't have carbohydrates around and your body starts breaking down fats like crazy, you get a buildup of a lot of acetyl-CoA, And this can be a bad thing in your liver. So your liver will convert excess acetyl-CoA into this molecule here, acetoacetate. And the acetoacetate gets metabolized into acetone and this other molecule here. But it's the acetone that's a ketone. Okay, it's in the name, ketone, acetone. Here's the ketone group right here. Okay, it's got that carbonyl that's bonded to 2 carbons, so it's a ketone group. Remember your ketones from Chapter 4. You can go look at your Chapter 4 table there. And so what happens is these ketones build up in your blood, and it's called ketosis. Okay, ketosis is it can cause an acidification of the blood, a condition called ketoacidosis. So this is pretty rare. It doesn't occur. So there's this one theory that like low-carb diets could cause this condition, this ketoacidosis, excessive ketosis or breakdown of, of fat and of acetyl-CoA. So a lot of like proponents that are against like low-carb diets say that it can result in too much fat breakdown, and too much fat breakdown can result in ketosis or ketoacidosis. But there's no actual cases of that occurring in people who were just like reducing their carb intake. However, it's a very real problem in diabetics, because in diabetics, in the case of diabetes, people with diabetes, their bodies, they break down carbohydrates into glucose, but they can't take up that glucose into their cells. So it's in their blood. They have tons of glucose in their blood, but it doesn't get into the cells. And where does glycolysis occur? It occurs in the cells. So if the cells can't take up glucose, they can't break down glucose, so they start breaking down fat like crazy. Okay? If you're on a low-carb diet, you're still getting some carbs into your cells. You can't really, it's really would be very difficult unless you ate only meat all the time. That's the only way that you could have a like carb-free diet. But so people who are diabetics, it's like their cells are carb-free, completely carb-free, so they break down fats like crazy, and they are the ones who are at risk of developing this ketoacidosis. Okay? So that's how we get energy from fats. Lastly, we can get energy from amino acids as well. Now, amino acids are really important for building proteins in our body, like enzymes like hexokinase and phosphofructokinase and lactase and sucrase, okay? All these enzymes that we've been talking about in the last couple of chapters. Proteins are very important in our body's functions. So we tend to use proteins in our diet. We break them down into amino acids, and then we use that pool of amino acids to build our own proteins. We don't really like to use those amino acids to make ATP. It's not their primary job. We have carbs and fats to do that. But if we get particularly desperate, if you're stranded on an island and you don't have any food and you're running out, your body's used up all the glycogen stores and it's using up, fat stores, and it really needs to get more energy, it can use amino acids. It can actually start breaking down your own body tissue. That's why you see like anorexics or starving, African children. They're skin and bones because their body has so little energy that it actually starts breaking down muscle tissue, because muscle tissue is very high in protein, and it's not like a vital tissue, except for your heart, which actually is how a lot of anorexic people can die in the end, is their heart becomes severely damaged by breakdown. So anyway, your body can use amino acids, and it can undergo that process of gluconeogenesis. I'm sorry, my PowerPoint keeps doing that. It undergoes that process of gluconeogenesis, right? Taking different amino acids and making glucose out of them. So this is a very detailed picture showing you the different all the different amino acids and where they can get fed in. So some of them, I should clarify, some of the amino acids can be converted into pyruvate, and that pyruvate can then be back converted into glucose. Some amino acids can actually be just physically sort of shunted into the citric acid cycle, and then they produce some of the products of the citric acid cycle, like the NADH and FADH2, that then get converted to ATP in the electron transport chain. So you have certain amino acids that can get shunted in there. Some amino acids can get converted directly to acetyl-CoA, which can be fed into the citric acid cycle, etc. So there's different amino acids can be converted into energy in different ways, either by being converted into glucose or converted into acetyl-CoA or some other component of the citric acid cycle. So you don't have to know which amino acids are converted to which. Just know that energy from amino acids comes from them being either converted into glucose by gluconeogenesis or converted into some molecule that can be fed into the citric acid cycle. Okay? So just a little, some fun facts about that is that we only get about 10% of our energy from amino acids. Most of it comes from glucose and fat. And that energy is extracted from proteins usually only when you're starving and you're not getting your energy from those other sources. So if your body needs to use amino acids for energy, a lot of times it has to delve into our body proteins. So technically our muscle tissue, usually the first place that it goes. So now putting it all together, hopefully your brain is not too addled by all of this. And if it is, maybe go take a break and then come back and watch this part of the lecture, or take a break and rewatch it again later, and it'll settle in better. But putting it all together, degradation of food biomolecules begins with digestion. Okay, so we digest things. Then, so those larger molecules get metabolized into smaller metabolites so they can enter the cells. And this is what this looks like. So this is from page 452 of your book. This is another sort of overview slide, okay, that kind of puts everything together. Your proteins, your polysaccharides, and your lipids, they get digested. Digestion is this process, the first step here, okay? Digestion results in these breakdown products, amino acids, monosaccharides, glycerol, and fatty acids are the different sort of breakdown products of these different large biomolecules. These large biomolecules, they can't get into the cell, right? They're stuck out here in your intestinal space, okay? In your, small intestinal space. Once they get broken down sufficiently, for instance, into amino acids, then into these building blocks, they can now be absorbed into the cells. Okay, so that's sort of step 2. Absorption of these hydrolysis products into cells. All right, and then we convert them into pyruvate or acetyl-CoA. These are the common metabolites, pyruvate and acetyl-CoA. So all of these things can be converted into one or both, pyruvate or acetyl-CoA. And then we have pyruvate gets converted into acetyl-CoA and acetyl-CoA. All this happens in the cell cytosol right now. We're in the cell cytosol. Conversion of any of these into pyruvate all happens in the cell cytosol. So what I wanted to say here, so this arrow here, converting monosaccharides to pyruvate, what is that reaction called? What do we call that? That would be called glycolysis. Glucose becoming pyruvate is through this blue arrow here. You can label it glycolysis. That's what that is. If we're turning pyruvate into glucose, what would you call that reaction here, going in the reverse direction? That would be gluconeogenesis, making something into glucose from some other product. So gluconeogenesis, this one could be called gluconeogenesis here. The breakdown of fatty acids, this blue arrow, the breakdown of fatty acids into acetyl-CoA. That is showing you beta oxidation. Beta oxidation is the breakdown of fatty acids into acetyl-CoA. So you could label that beta oxidation. What else? That's about it. So those are the ones I want you to label there. And this is all taking place in the cytosol. Then pyruvate gets converted into acetyl-CoA, and acetyl-CoA can get transferred into the mitochondria. So now all the reactions that occur, the citric acid cycle, the electron transport chain. Now we're all taking place in the mitochondria of the cell. So that acetyl-CoA, whether it comes from proteins, glucose, or fatty acids, okay, gets shunted into the citric acid cycle, okay? And the citric acid cycle produces carbon dioxide as a waste product. It also produces a lot of these electron carriers, the NADH and FADH2 molecules, okay? And those electron carriers then proceed to the electron transport chain, where they undergo where the reaction of oxidative phosphorylation occurs. You get those electrons get dropped off, they pump those protons, and then when the protons come back in, they get oxidized, which is very exothermic, and that energy from the oxidation of those protons is used to make ATP, to make lots of ATP. Oh, so the pink and blue arrows are showing you catabolic reactions, so breakdown reactions, versus anabolic reactions, the build-up reactions, building up. So, another way. So this one here, taking monosaccharides and doing an anabolic reaction of building polysaccharides, that would be glycogenesis, making glycogen, okay, from monosaccharides to polysaccharides. So taking glucose and building it into glycogen would be an anabolic reaction. The yellow arrows are showing you the flow of electrons. So electrons being captured by these electron carriers, NADH and FADH2. Also, when you undergo beta oxidation here, you produce some NADH and FADH2. Sometimes breaking down amino acids can produce NADH or FADH2. Okay, so these are all the different places where these electron carriers can come from. So it kind of puts it all together for you in terms of catabolic reactions, anabolic reactions, and where sort of electrons come from. So that first slide, initially I had a slide that sort of showed you in a little bit of detail this first up to the citric acid cycle, but it kind of left off the electron transport chain. So this one incorporates that. So the byproducts of the oxidative phosphorylation are ultimately ATP, lots of it, depending on how much NADH and FADH2 you have, and also water. Remember, water is a product of that oxidative reaction. And that's why you need oxygen. That's why we breathe oxygen, because if we don't have oxygen, we can't undergo oxidative phosphorylation. I sort of forgot to mention this on the other slide. Oxygen is very important for making ATP. You can't make ATP, you can't make significant amounts of ATP without oxygen, and especially in your brain that relies on glycolysis and oxidative phosphorylation to make ATP. Your brain cells don't function very long without oxygen. They don't function very long without ATP, and they can't make ATP without oxygen. So that's why it's so critical that we breathe oxygen, because without it, we can't make ATP, and without ATP, our body can't fuel chemical reactions, and we constantly have chemical reactions going on in our cells to keep them alive. It's like cutting the power, okay? You cut the power, and all the electricity goes out, and you can't run your microwave, you can't run your TV, you can't run, all the electronics in your house. Same thing. If you don't have any oxygen, you can't make ATP, you can't power the activities of your body, even the most basic ones like your breathing and your brain function. If your brain can't function, then it can't regulate your breathing and your heart rate, and eventually, so the longer your brain is oxygen deprived, the greater the damage done to your body and to your brain. So that is how we get ATP, ultimately from proteins, carbohydrates, and lipids. Ta-da! I know it's a lot. It's kind of like a lot of things to remember, mainly all the stuff that we talked about with carbohydrates. But reread it, rewatch the lecture, and hopefully it'll all make sense to you. And if it doesn't, let me know, or better yet, contact your local tutors and work through it together. And but also let me know if there's parts that you're having trouble with. But again, just probably reading through it a couple of times or watching the lecture a couple of times will really help. I think it's just can be a lot. It can be an overwhelming amount of information, but it's trying to keep it organized in these separate sort of groups. So that's all. Chapter 12, Metabolism. Hope you enjoyed. That's my last lecture. Bye now.